All right, so today we are going to review Chapter 7, which is the unit on cellular cells and what comprises of cells. The first slide I got up here is really the difference between prokaryotes and eukaryotes. And what you can see here is that about 4 or 5 things are the same between prokaryotes and eukaryotes. Obviously, prokaryotes were the primitive cell, or what came before eukaryotes, because they have a lot less stuff inside of them. So, as you can see, the plasma membrane, which is right here, the cytoplasm, which contains a cytosol, is in this area. The DNA, or the chromatin, is in the middle of the cell, and that's where the nucleoid region is in prokaryotes. And then prokaryotes also have ribosomes, which occur in both prokaryotes and eukaryotes, but that's about the only similarities between the two. Not every prokaryotic cell has flagella, and very few eukaryotic cells, if any, have flagella. So those are the things that pretty much comprise of both prokaryotes and eukaryotes. One thing in particular that both of them contain are ribosomes. And ribosomes are made from the nucleolus. And within that nucleolus is a large non-dividing unit of DNA that comprises of rRNA subunits, which come together to form these ribosomes. So this is kind of a molecular look at what a ribosome looks like, but again, ribosomes are just the 2 large and small subunits that come together to create proteins or transcribe them off DNA. We'll talk about that a little later on in the year. Now, this picture is showing just a diagram of the nucleus and the endoplasmic reticulum. This endoplasmic reticulum is the rough endoplasmic reticulum, and that's mainly due to the fact that ribosomes cover the surface of the ER. To start out with, though, the nucleus here contains chromatin. Sorry, I ran out of room there. Chromatin, which comprises of mainly the entire nucleus. Remember, it's unconcentrated DNA, which actually binds up to form chromosomes. So don't the DNA or genetic material that's inside the nucleus is not always in the form of chromosomes, but in this picture is in the form of chromatin. After being transcribed into RNA segments, the RNA actually snakes out of these nuclear pores. And when it comes outside of the nuclear pores, what it's going to do is run into a ribosome. That ribosome can either be a free ribosome, which is just floating around in the cytoplasm, or it can be a ribosome that's bound to the endoplasmic reticulum. If that happens, if the RNA actually comes in contact with one of the ribosomes on the endoplasmic reticulum, it's going to synthesize into a protein right there, and the protein is going to go back into the ER, which is going to then snake it through the rest of the system. This is known also as the endomembrane system, which really transports DNA into proteins and then buds the proteins off the ER to facilitate getting to the membrane of the cell. So this entire endomembrane system, which is connected, transports the RNA to the ribosomes, which transports the protein to the next part we're going to look at in a second here. Before we leave, just one last thing here. The nucleolus, again, is right in the center of the DNA. Just because it's in the center doesn't mean it holds all of the most essential information, but that just represents the non-dividing part of the nucleus that forms the ribosomal subunits. The next part we're going to look at here is the next piece of the endomembrane system, which is the Golgi apparatus. After proteins are synthesized and then transported through the endoplasmic reticulum, they bud off of the system in little vesicles that arrive on the cis side of the Golgi apparatus. When they get to the Golgi apparatus, they get modified and packaged up into what the final protein is going to look like. And after that packaging occurs, are budded off the trans side of the Golgi towards the membrane. Most proteins that are synthesized from the endoplasmic reticulum are glycoproteins, which, after coming off the Golgi apparatus, form those glycoproteins and lipids that encompass the cellular membrane, which helps the cell communicate with other cells and move around. So this entire process of the endomembrane system and the functioning of the Golgi helps create these proteins that either get expelled outside the cell or function on the surface of the cell. One of the last pieces of the endomembrane system we're going to talk about are lysosomes and peroxisomes. Lysosomes are made in the same way as peroxisomes, and they are enzymes that break down food particles or damaged organelles or anything like that inside the cell. They do that by hydrolyzing the food particles, which means breaking them up by introducing hydrogens to the food particle or whatever it's breaking up. As soon as that's broken up, the food can be used in cellular respiration for energy production, and the Golgi buds the lysosomes off of itself from the cis side, which then float around and look for food particles to digest. The next piece we're going to talk about is the cytoskeleton. The cytoskeleton comprises of microfilaments and intermediate filaments that make up the inner workings or the inner structure of the cell. These are where the microfilaments are. The microfilaments provide structure, which usually make up of the actin filaments inside the cell. The microtubules also provide structure and transport around the cell. Those are come out of the centrioles, which are located in the centrosome of the cell. Microtubules can be formed and destroyed spontaneously, so new paths are always being formed around the cell. The spaces between the cells are known as gap junctions. Okay, and gap junctions is actually an animal cell term. If we were talking about plant cells, these openings would be the exact same openings in between cells, only they would be called plasma desmata. And what they do is they facilitate communication between the cells. There are micropores that help chemicals get between cells very quickly. A few other connections between cells that we'll talk about are tight junctions, which actually block the passage of fluids between the outside layer of the cells and the inside layer, and desmosomes, which just facilitate the connection between cells. We will talk a lot about these next two pictures in the next unit we're going to do, which is on cellular energy. But just note that mitochondria and mitochondria and chloroplasts facilitate cellular production or cellular energy production, and mitochondria are the organelles that go through cellular respiration, whereas photosynthesis is undergone in the plant cell or the chloroplasts. So that's really the difference between those two right now. We'll talk about those a little more in particular later on. And the last bit of information I want to give to you guys before this is over is the endosymbiosis theory. Okay, and in the endosymbiosis theory, what was basically come up with was that in an ancestral cell, which really didn't have organelles in it, the ancestral cell in devouring or engulfing prokaryotic cells actually formed these organelles. So in the ancestral cell, we have no organelles. And as organelles or prokaryotes get engulfed into the cell through millions and millions of years of selection, are selected for prokaryotes that actually do one job really well. And then as cells divide and proliferate, keep those organelles within and are able to do multiple number of jobs at the same time. These, in being prokaryotes at one point, these mitochondria and chloroplasts still contain their own DNA, so they divide on their own and within the cell operate on their own. So that is your review of the Chapter 7 cell unit. Thanks for listening.